my name is Molly Kozminsky, and I'm a third-year graduate student in the Department of Chemical Engineering. Today, we're going to talk about how we use my research to solve problems faced by clinicians when it comes to diagnosing and studying cancer. It is hard to understate the influence of cancer and all of the people that it has affected. Cancer is the second leading cause of death in the United States, killing over 1,600 people every single day. But not all cancers are created equal. It turns out that over 90% of cancer deaths are caused when the disease spreads. When you hear people talk about breast cancer or prostate cancer, they're talking about the primary tumor. This is where the cancer starts. However, of the many cells in this primary tumor, a select few of them are able to begin, and even fewer still will be able to complete, the multi-step process by which cancer spreads, which is known as metastasis. The first step of metastasis is for a cell to leave the primary tumor and enter the bloodstream. From there, fewer still of those cells that are able to leave the primary tumor are able to migrate and survive in the bloodstream. Of those, even fewer still are able to leave the bloodstream, and fewer still are able to colonize a secondary location. You can see how it's important for us to be able to identify and study those cells that are able to complete this metastatic process. Now, clinicians already do have a technique to diagnose and study cancer, known as the biopsy. However, there are several issues associated with the traditional biopsy. First, it can be a highly invasive and painful procedure, causing a great amount of discomfort to the patient. Depending on the health and disease status of that patient, it might not even be possible. Two other associated issues with the traditional biopsy are somewhat related. One is that of underdiagnosis. If the traditional biopsy is not sufficiently sensitive, it may miss the disease entirely. Another issue is that of overdiagnosis, which means the biopsy might diagnose a disease where the disease itself would cause less pain and discomfort than the treatment. However, there is an additional issue associated with the traditional biopsy, and that is that it doesn't tell the whole story. Take a look at this picture. This is a picture of a prostate biopsy, and as you can tell from looking at it, there are two different regions. If you look to the left of this black line, you see a lot more of those dark pink circles, and they're packed more tightly together, and there seems to be like there's a bit more structure. However, if you look at the right of that black line, you see a lot more of that light pink. It seems a lot looser, and it's not terribly packed. The difference between those two regions is how seriously they've been scored based on the seriousness of the disease. Those on the left are actually scored to be less serious than those on the right. However, if the needle had been moved a bit to the left, we would have completely missed those cells that would have been more indicative of a more serious disease. This highlights the need for a more informative diagnostic technique. Given the drawbacks of the traditional biopsy, it is imperative that we develop a new diagnostic technique that is simultaneously somewhat non-invasive, sensitive, and informative. To do so, we can look to the method by which the cancer cells spread. Having already left the primary tumor, those cells in circulation, or circulating tumor cells, known as CTCs, present an opportunity to scientists and clinicians. They are accessible by a simple blood draw. However, there are two challenges associated with the study of CTCs. The first challenge is that in a couple of drops of blood, there are billions of surrounding blood cells, cells that are not the cells we're interested in studying. The second challenge is that there are very few of these circulating tumor cells with which to work, meaning depending on how many different types of study we'd like to conduct, we need more material. To overcome these challenges, we need to assemble a team of clinicians, biologists, and engineers, each bringing their own expertise to the table so that we can best study these cells that are at the root of cancer mortality. So at this point, you might be wondering, what is a chemical engineer, and why would a chemical engineer be involved in medical research? Well, to talk about what a chemical engineer is, it's easiest to talk about what they do, which is separation and scale-up. To talk about separation, think about oil. When you first take oil out of the ground, it's not terribly useful for anything. What you need is for a chemical engineer to come in and do a distillation process or separate it into its components. And then you have all different components that are useful for many things, such as kerosene or gasoline for your car. When it comes to scale-up, think about aspirin. When aspirin was originally synthesized, it was in a lab somewhere, and the chemist originally probably didn't make very much. But then think about how many aspirin tablets are taken every single day. We need more of it. And that's where a chemical engineer came in. and developed a process that made lots of aspirin. So, what does all this have to do with circulating tumor cell research? Well, it turns out that as a chemical engineer, this is exactly where I come in, and this is exactly what I do. I start by separating the circulating tumor cells from all of the surrounding blood cells, which is a separation process. Then, because there are so few of them, we need to scale them up, and I work to grow them so that we have more material. So how do we capture these cells and isolate them from the surrounding normal blood cells? Well, it turns out we have to think small, like really small. We need to get on the cellular level in order to overcome these challenges. And the way we do this is by using two different length scales. So we use microfluidics, or very small channels, channels so small that they're only the height of a human hair, or just one of them. Then within these small channels, we have nanomaterials. And these nanomaterials are on the scale of 1 nanometer. And that's about the same size as a base pair of DNA, which is so small that it's actually already inside of cells. So we make microfluidic devices, using these microchannels to enclose assemblies of those nanomaterials. On the nanomaterials, we put molecules that stick to the circulating tumor cells, but not the white blood cells. So what happens is we flow the blood through these microchannels, the circulating tumor cells stick to the molecules on the nanomaterials, and then the white blood cells flow through and out the other side. Once we capture these cells, we can grow them up, so we have more material to study. This is what we're working on now. If we're successful, we have a lot of different options for analysis. We can look at the different proteins that are expressed by the cells, or we can look at changes in their DNA, which could help us predict which drugs would work best on which patients. One of our long-term goals is to be able to test therapeutics on the cells before they would even reach the patient, so we could see if a particular existing drug could possibly work for a patient, or we could even test new therapeutics. Because what this is really about is the patient, and bringing new technologies from the benchside to the bedside. Clinicians and engineers can work together to develop a sensitive, informative, and non-invasive diagnostic technique that can yield new information. The results of such analysis can then be brought back to the patient to improve his or her treatment. And that is how chemical engineers can contribute their expertise to developing cancer diagnostics. Thank you.